Thanks, Michelle. And hello, everyone. I hope we're all sort of well fed and well watered after lunch. I'm privileged to sort of have that after lunch slot, because I know there'll be no question. So, as Michelle said, I'm from the Parkinson's UK Brain Bank. My name's Rory. I'm the tissue coordinator there. For those who don't know anything about what the Brain Bank might entail, I'm going to run through that today. But to sort of say that I'm the individual at the Parkinson's UK Brain Bank who helps facilitate the donations of brains to research, as it were. So today, I thought I'd keep it simple with sort of three things, saying who we are, what we do, and our process. So to clarify, sort of, I'm working at Imperial College in West London, and we're a joint effort and venture between the MS Society Tissue Bank and the Parkinson's UK Brain Bank. So we're both focused on neurological conditions that occur in the human brain, and so that's why we've joined efforts to make the best use of our resources to sort of become the Multiple Sclerosis and Parkinson's Tissue Bank based at Imperial College. So with three entities wrapped up in one to do this, we're a team of neuropathologists, technicians, and sort of run-around staff is what I call myself, okay? What we do, as you sort of gathered from that opening, the main part that we do is we fulfill people's wishes. A lot of people want to donate to research. This type of donation is separate to sort of NHS organ donation, and it's separate to whole body donation. With those two schemes, people with neurological conditions may often be ineligible for those. But there's a desire to sort of donate to research and help further understand the conditions that people suffer from, and that's what we do. So we facilitate the donation of the brain and the spinal cord, so not whole body donation as it were, and we're what's called a research tissue bank. So we don't collect things to sit on a shelf, we collect things to help support research. And so what we're able to do is expedite research, because people can apply to us and we can provide ethical permission from a board that includes people living with Parkinson's, experts in the scientific field, and lay members to sort of review these applications and make sure they're put to the best interest. Similarly, we work with other cohorts and other studies to ensure that we're sort of closing the gap and performing a clinical audit on what people may have participated in through research or may have experienced through their lifetime, to understand how that is reflected with changes in the brain. So far, with the Oxford Discovery cohort, 165 people have signed up. We've been able to facilitate 28 donations from individuals within this cohort, and we're working with Annie, who will be presenting after me, on sort of doing some research into those samples with a very rich set of data that's been provided by the team at the Discovery. So I've mentioned sort of what we do about that donation process. Obviously that's our main focus, but we sort of have four different objectives. One is, as I mentioned, we speak with people who want to donate their brain to research, but we speak with their relatives as well, and we're able to provide closure with producing a neuropath report for them. That can occur within 6 to 12 months after an individual passes away, and that can often assist with sort of understanding exactly what may have been occurring in someone's lifetime. I've touched on that we work with researchers as well, and so we look after this process so that it enables research to be done as quickly as possible. But beyond that, we work to educate as well. We want to be an educational resource for individuals, and that's really what and I'll touch on our sort of 5-year plan in a second. What we're trying to focus on is educating health professionals, so we often host visits from Parkinson's specialist nurse and MS specialist nurse, and we provide an educational resource for people undergoing medical training to better understand neuroanatomy. But furthermore, what we're doing is supporting a lot of research, and we want to enable people to be kept up to date with that. And so what we do is we work with Parkinson's UK, who kindly fund us, and part of this 5-year plan is with reviewing these applications is to make sure that we're making these available for individuals like yourselves to see what we call the basic research that's being supported. Why is a brain bank necessary? What do we do, sort of thing? The reason why it's so important, and with what's different sort of observational studies, perhaps with blood banks and stuff like that, where you might be able to understand what's going on there, as long as everything's gone well, your brain will stay with you until you pass away. And once, pause for laughter, hey. But once that donation has occurred, what we're aiming to do is to take information from people's medical records, information from people's questionnaires, whether that be their symptoms or sort of their family history. Bring in, when we're sort of working with cohorts like the discovery cohort, all that rich information that they've collected about a condition, and then to say, okay, let's bring all this together. Let's have a look at the brain, see what we can observe, and then sort of draw these links on a most basic level. Of course, we do a lot more than that, but that's what the tissue bank is there to enable, is this collation of information from the medical practitioners, from yourselves living your own sort of personal experience, and what we find in the tissue. So you're probably thinking, oh, this all sounds great. What do I do? How do I get involved? This has sort of been the theme with these talks. And with people sort of signing up to sort of record their wishes to donate their brain, they do that, as always, through a set of forms that you can get from us by calling us or sort of emailing us. We have a set of information leaflets and forms around today, if anyone's interested. And then, as was touched on in an earlier talk, what we're now looking to do is for people who sign up with the tissue bank is to undergo sort of questionnaires, which we're currently doing yearly to every 2 years, but also work with that Cognitron platform. So we can really track there with sort of how cognition is going, and then see how that reflects in the brain donation later on. And of course, we nag you with newsletters and websites and through social media. You're always welcome to visit the tissue bank. You can get in contact with us, and we often sort of try and be out in the public so people are aware of what we're doing. And just to sort of speak on what the process is of donation. We're a dedicated team. We're set up thanks to Parkinson's UK and thanks to the MS Society, that we look after that entire donation process. Of course, we work with local hospitals, we work with local GPs and funeral directors to enable that, but we're the team that sort of coordinates that, and we don't look to add any more stress to families or what must be a difficult time. So when someone passes away, we're called, and then we look after all further arrangements. So that be sort of making sure that certification is in place, speaking with local hospitals and the mortuaries there, enabling transport of individuals to the hospital to have that donation occur, and of course then that step of the brain tissue collection to then be taken to the laboratory at London. Happy to speak more on that a little bit later. But sort of what I wanted to speak about is every donation that we receive is vital, and it's a unique decision for each person who decides to donate, because they may not feel that they can, or they may feel that there's certain things that stop them from doing so. So anyone who does feel that they can donate their brain is incredibly important. We're not always able to facilitate these donations. It says it has to be done within 48, 72 hours. And donations from every individual, whether that be someone living with a neurological condition or otherwise, is very important to understanding that the basic processes of the brain, being able to bear comparisons between different people, as this isn't something that's routinely done. We're sort of acting as an extension of the NHS service by providing this. And of course, each donation is carefully examined and a neuropath report is produced. By way of introductions, we're a big team down at Imperial. We have Professor Steve Gentleman and Professor Richard Nicholas sort of running the scientific direction of the tissue bank, making sure that we're sort of kept up to date with techniques at all times to make sure that we're preserving tissues in the ways that are most suitable for research. And we've got a set of neuropathologists, some more camera shy than others. And myself and Dr. George Gevrich, who I'm sure many of you may have met in this room prior, along with a massive technical team of 4 technicians who are sort of handling each donation and making those prepared for research as we go. So that's me. I'm Rory from the Brain Bank, and I sort of hope I've been able to sort of speak a little bit about the toolbox of which Parkinson's UK is providing to sort of assist us in both furthering basic understanding of Parkinson's, but also providing all these sort of clinical trials that are occurring as well. So we're one part of that toolbox. I'd like to sort of pass over to Annie, who is doing work on these very samples that people have generously donated.
Thank you, Rory. Hello, everyone. My name is Annie, and I am a final year PhD student with Professor Rory Parkinen. And our group studies the neuropathology of Parkinson's disease, and we work on a lot of post-mortem brain samples, gut biopsy, and then stool samples from PD patients as well. So for my study, I have been working with, I've been acquiring post-mortem brain tissue from Imperial Brain Bank for the study of pathology in Parkinson's disease using this very novel technique called spatial transcriptomics. I'm going to start with introducing you a bit about the neuropathology of PD. So as you may know, the reason why there's no treatment for PD is that we don't understand the disease progress enough at the moment. So with years of research, what we know so far is that we see this loss of selective loss of dopaminergic neurons in Parkinson's disease brain that mainly happens in the substantia nigra. This is actually quite obvious if you just take a postmortem brain sample tissue and look at it. So this picture here shows that the midbrain and the black region here is the substantia nigra. So it's because the dopaminergic neurons are actually pigmented. So as the disease stage progresses, you can see that we're losing this pigment in the substantia nigra, and that is actually indicating that the dopaminergic neurons is being lost. Now, this cell loss actually also happens in many other regions in the brain, and also being observed in many other cell types that's other than just dopaminergic neurons. And at the moment, it's really unclear how this cell loss occurs and why, what makes these dopaminergic neurons more vulnerable to the cell loss or the pathology than the other neurons. And it is believed that the death of these neurons is caused by the aggregation of this protein called alpha-synuclein. And when I say aggregation, it means that the protein itself actually misfolds to make a clump in the brain. And that's what actually we think is causing the neurons to die. And this is in turn called Lewy pathology. Now, if you look at these two pictures here, these are actually taken from the same substantia nigra, from the same PD brain sample. And on the left here, this is a neuron without pathology, so it's actually a healthy-looking neuron. So you see the cell body with the nucleus here, and the black bit is the pigment of the dopaminergic neuron itself. And on the right here is the neuron with Lewy pathology. So how we visualize this clinically is to take a piece of brain tissue and stain it with the antibody that targets the alpha-synuclein. So I don't know if you can see it clearly at the back. So there is a round light brown structure here. This in the middle of the neurons is the Lewy pathology. So we think that eventually this neuron is going to die at the later stage of the diseases. Now, another thing I want to mention is that within the brain, the Lewy pathology is thought to progress to spread by this Braak staging. So it's start from the brainstem of the brain, and then slowly progresses to the midbrain, and then in the later stage of diseases to the cortex of the brain. However, the truth is that many PD cases, when they are diagnosed, they're already at stage 5 or 6. So we don't really know what happens in the earlier stages of the disease because there are many outliers to this scheme. So some of the cases don't really follow this pattern. Now, with those questions in mind we have about neuropathology of the PD, so our approach is to tackle the problems on a genetic level. So the nucleus of a cell actually contains all the genetic information, everything you need to know about this cell, this neuron. And for us to understand what's the difference between the neuron with the pathology and the neurons without pathology, is simply we can ask the question, what's the genetic differences between these two groups? And then if we do find a genetic driver that's causing these neurons to take up the Lewy pathology than the others, then these genetic drivers can then, we can then use them to be potential future drug targets. For example, to prevent the alpha-synuclein to aggregate inside the neurons, so therefore to prevent or cure the disease. And so there are many ways that you can get the genetic information from your sample. So I'm going to try to explain this. I actually brought some tools with me today. So I'm going to start from box sequencing. So how we carry this out is that we have a little tube that's seen commonly in the lab, and then we're going to take a little bit of the brain samples and put into this tube, and then fill up with buffer. And then we're going to just use a magnetic bead to mash everything together, so the cells then releasing the genetic information from your sample. So we now have a little tube containing the genetic information from a sample. This will be sample sequencing. So if you actually imagine it, it's more like making a smoothie, that you're blending everything together, and then you can get the flavor of the smoothie, but you don't actually know what the flavor is coming from, which cell type. Okay? So in this term, that you're getting all the genetic information from your sample, but you don't know what cell types the sample is containing. So this technique can be useful, for example, if you want to compare the gene expression information from a PD sample to a control sample to see what the difference is. That's what the disease is causing the brain to have. And then it comes to single-cell nuclei sequencing, which is developed around 2012 and still used quite extensively at the moment for research. And you can think of it as a fruit salad, that you can see now what type of fruits there are, but then even though everything is still blended in together, but you can get to see, it still happens in the tube, but you can get to see what's the cell type that's contained in your sample. So you can try to study what's the cell type proportion in PD sample compared to control sample. So you get a more refined information from this technique. And then it comes to the spatial transcriptomics that we've been using. It's developed really recently, and then we're trying to optimize this technique on human brain tissue at the moment. So as the name suggests, it has that spatial information. Okay? So if you imagine it as a fruit tart, you can see where your fruits are now. So instead of happening in a tube, and I have a glass slide that we use it a lot in pathology labs, basically, to identify the pathology. So what we're going to do is we take a really thin layer of the human brain tissue and put it on top of the slide, and then we're going to stain it to visualize your protein of interest. So in this case, we're going to visualize the neurons with and without pathology, okay? And then, so now we're not only getting the gene expression information from these cells, but we also get to see where the cells are located. Okay, so we get that spatial information. We're going to analyze, we're going to manually select these two types of neurons and then analyze them and compare the gene expression profile. So I got a picture here that's actually coming from the spatial transcriptomic technique itself from the machine. So how we start is that we put the tissue on the slide and then stain it with the protein you want to visualize. And then, so in green here is the dopaminergic neurons. So actually, sorry, I forgot to mention, this graph is coming from this one substantial diagram of one PD brain patient, this PD patient brain. So in green here, that stands for the dopaminergic neurons. You can see 1, 2, 3, 4, 5, 5 dopaminergic neurons in this field of view. And in bright red or orange is the Lewy pathology, okay? And if you look at this neuron here, and that is the dopaminergic neurons containing the Lewy pathology. And right next to it is the healthy-looking dopaminergic neurons that don't have the pathology. So even though they're so close together, you can see this neuron is affected by the pathology, but this one doesn't. And why is that? We don't know. So we're gonna, the next step is to manually select this neuron and that neuron, put them into different tubes. So the machine is gonna then extract the gene information from these two types of neurons. And then this will be sent for sequencing, and then with the gene expression profile of these two groups can be compared. So I got a bit of data from our pilot study. So this is from one PD brain sample. And I'm going to try to explain this plot to you. So this is actually called a volcano plot. So at 0 here, this is the baseline, okay? And you can see there's many small dots, and each dot represents one gene that's coming from our human body. And the point is that the more far apart, far away from the baseline these dots are, so these ones are labeled genes, the more different the gene expression level is between the neurons with and without Lewy pathology. So these are the genetic driver we're looking for that's causing the disease, causing them to change. So the plan is at the moment is that we're going to do this experiment for 15 PD and 15 control brains, starting from the substantial nigra and then proceed to other brain regions that's affected in PD. And then hopefully, if we do validate this finding, we're going to find it's going to help with the development of new target for gene and drug therapy and help us to understand the disease progress better and help you better. Okay, and that's the end of my talk. Thank you very much.